The study of cells is called cytology. A cell is a unit of life surrounded by a semi-permeable membrane and capable of reproduction in an environment devoid of other living things. Cells vary tremendously in size, shape, and function. Dozens of different types are present in the body. Some, like those lining the intestinal lumen, divide every few hours to replace those damaged or lost through normal processes. Others, such as bone-forming cells, divide much less frequently. Nervous, skeletal muscle cells, and red blood corpuscles do not divide at all once they reach a mature stage. Cells multiply by dividing, and this typically results in two daughter cells that are identical to the parent cell. Under certain conditions, the daughter cells can evolve into cell types that are different from that of the parent cell, a process called metaplasia.
Hi, I'm Mike Shively, and welcome to Introduction to Anatomy and Physiology. Our subject today is cytology. Now, we all know that the suffix ology means study of, and the prefix cyt, C-Y-T, refers to cells. So this is the study of cells. In the last century, actually 2 centuries ago, a couple of Germans by the name of Schleiden and Schwann promoted what has come to known as the cell theory. And the tenets of the cell theory, if we can go to our overhead camera for just a moment, the tenets of the cell theory are that all living things are composed of cells and their products. And secondly, that barring initial creation, cells are derived only from pre-existing cells. This basic theorem has been tested and retested and basically is held to be absolute truth today. All living things are composed of cells and cell products, and cells are derived only from pre-existing cells. In other words, you and I couldn't go down to the Napa cell parts store and pick up a bunch of cell parts and actually construct a cell this afternoon because we don't have the ability to do that. If we were to take a look at the DNA in any one of our body cells, it doesn't make a difference what kind of cell it is, what would be peculiar about it in reference to any other cell from another part of our body? It would be different. Actually, it'd be the same, would it not? It would be the same, barring some possible mutations. So even though we've got many different kinds of cells in our body, matter of fact, several dozen different types, all of them have the same genetic makeup. They all have exactly the same DNA. Let's take a look at some examples of some of the kinds of body cells that we do have. Now, these are just a few examples, so we'll look at this first graphic. We know that neurons are the cells that conduct impulses. RBCs, or red blood corpuscles, are involved in oxygen transport. Skeletal muscle cells, of course, contract. We have some secretory cells called goblet cells that secrete mucus. Osteoblasts are bone tissue forming cells, and fibroblasts are cells that manufacture various kinds of fiber proteins. Now, there are dozens of other kinds of cells in our body in addition to these, but these are just some examples. And remember that all the cells in the body have the same DNA, barring some possible mutations. And so how is it that we can have so many different kinds of cells if they all have exactly the same genetic complement? How can we have dozens of types of cells? Adam?
Only part of the DNA is triggered to produce proteins that differentiate the cell.
Exactly. Even though all the cells have the same DNA, not all of the DNA in a given cell is used by that cell. So skeletal muscle cells, for example, use parts of the DNA that are important in producing the particular contractile proteins that are necessary for their function. And pancreatic cells that produce pancreatic juice may use some of the same parts of the DNA as well as additional different parts. So that's how we have different kinds of cells, even though they all have the same genetic makeup. In order to really understand cytology, I think we need to go down to the subcellular level for a while in today's session and spend some time talking about the cytochemistry, really. And so if I were to ask you, what are the most common elements in the body, what would you think? What's one of the most common? Susan?
Oxygen.
Oxygen is certainly a very common element in the body. Another one?
Carbon.
Carbon, Adam, that's good. Tyler?
Nitrogen.
Nitrogen. Amber?
Hydrogen.
Okay, let's take a look at these. Let's take a look at a graphic that shows the distribution of these four elements in the body. Oxygen makes up by weight, now this is by weight or mass, oxygen makes up about 65% of the body mass, carbon about 18%, hydrogen 10%, and nitrogen about 3%. Now if you add up those four elements, that takes care of about 96% of our body weight. So even though we have about 100 plus or minus elements that are present on Earth, four of them make up over 90% of our body mass. Now the remaining 4% is actually, most of that is about 8 or 10 others, with only very tiny trace amounts of a lot of other elements as well. Moving beyond the elemental level, we'll talk about compounds and go to the overhead camera for just a moment. The most common compound in the body is what? Water. Now water is a relatively simple inorganic compound, has 2 hydrogens and 1 oxygen atom making it up. And this particular compound is about 2/3 of our body weight. That means if you weigh 150 pounds and we put you in a oven and dry you completely out, desiccate you completely, of course you'd be dead, but the fact of the matter is there's only really 50 pounds of other kinds of things in your body if you weigh 150. 2/3 of the body weight is water. And then there are other inorganic compounds as well as a lot of simple organic compounds within the body. Let's take a look at a graphic that will look at some of the simple compounds that are present in the body. We've mentioned water, and of course there's a lot of carbon dioxide and many other simple, relatively inorganic compounds. But we've got simple organic compounds like amino acids, simple sugars, fatty acids, and nucleotides. And those simple organic building blocks are present in and of themselves within our body, but in many cases they are polymerized to form what we call biological macromolecules. And you are probably aware that if you hook amino acids together in a continuing chain, you eventually will form very long polymers of amino acids that we'll call polypeptides, or even if they get long enough, we'll call them proteins. If you hook simple sugars together, you'll end up with very long polymers of those simple sugars, end up with a compound that we'll call a polysaccharide. If you hook certain kinds of fatty acids and glycerol together, you may make glycerides, one of several different kinds of lipids. And if we hook the building blocks called nucleotides together, we'll end up with what we call nucleic acids. Let's look at a graphic that shows the 4 major classes of biological macromolecules that we just mentioned. Proteins, of course, formed of amino acids. Polysaccharides, formed of simple sugars. Lipids are actually a very diverse group of biological macromolecules, including several different compounds that we'll look at very briefly in a bit. And the nucleic acids that are probably more familiar by their acronyms, RNA and DNA. Does anybody here know what RNA actually stands for, the long version? Adam?
Ribose nucleic acid.
Ribonucleic acid. RNA is an acronym for ribonucleic acid. And DNA is an acronym for what? Hazel?
Deoxyribonucleic acid.
Deoxyribonucleic acid. Those are the two nucleic acids that are very important in cytochemistry. Let's take these biological macromolecules and talk about them individually for just a moment each. And we'll begin with proteins. And when we think of all the kinds of things that proteins do, it would probably be easier to make a list of the kinds of things that proteins don't do, because proteins are extremely important in body structure. Not only do they form structural components of cells, but they also are involved in antibodies, they're involved in enzymes, they're involved in hormones. Many of the hormones are proteins. As a matter of fact, there are more hormones that are protein than there are hormones that are made of steroids and the other kinds of compounds that are involved. We know that the oxygen-binding pigment in red blood corpuscles is a protein, has a protein component. We know that the contractile elements in muscle tissue is protein as well. Let's go to the overhead camera for just a moment and take a look at what the basic building block is. An amino acid is a relatively simple organic compound that has this basic structure. We have a carbon atom that is bound to a amino group, an NH2 group. It's also bound to a carboxyl or organic acid radical. There's a hydrogen atom on that carbon atom, and then there's something else. You know, chemists use the R symbol for something else. And the thing that makes one amino acid different than another amino acid is what forms this R. In the simplest amino acid, of course, it's just another hydrogen, but it can be a lot of other different things. And by replacing this R with different things, then we end up with about 20 different amino acids that are found in the body. And it turns out that about 8 of those are considered to be essential amino acids because they cannot be synthesized by our body, and we must intake those in our diet, in the foodstuffs that we eat. The other amino acids that are found in the body, we don't have to have in our diet because our body can make them from other products and perhaps form them from other amino acids. Now, if you hook amino acids together, then you form a combination of 2 and then 3 and then 4 and so forth, and you do this with a process that you're familiar with from basic biology called dehydration synthesis. And we end up with a big, long string of amino acids that eventually, too many to conveniently count, and we call that a polypeptide, or if that polypeptide chain is more than 100 amino acids long, then we call it a protein. And again, the proteins serve so many different functions in the body. Let's take a look at a graphic that shows some of the roles that they play. Again, we mentioned these before, perhaps, at least some of them. There are structural proteins. The antibodies are proteins, enzymes, hormones, oxygen-binding proteins, both hemoglobin and, of course, myoglobin in muscle tissue as well. And then there's contractile elements in muscle tissue that is formed from protein as well. If we take a look at the next graphic, we can see specific examples of the kinds of things that we're talking about. The structural components, fibers are collagen, elastin, and keratin, or at least examples thereof. Antibodies are also known as gamma globulins. Those are defensive proteins that are involved in the immune process. Some of the enzymes, examples include, pepsin, trypsin, amylase, and a number of others as well. And I mentioned that the majority, in fact, of hormones are formed of proteins. Here's just 2 hormones as examples, insulin and parathyroid hormone. Oxygen-binding proteins include the hemoglobin and myoglobin, and then contractile proteins, among others, include actin and myosin. So proteins play many, many functional roles in the body in many different kinds of body cells. They're extremely important in all body cells, but of course specialized cells will use different ones of these to various degrees. Carbohydrates, if we could go to our overhead camera for just a minute. Carbohydrates, we often acronym CHO''S like this, just short. These are chemical compounds that have carbon and hydrogen and oxygen present in a 1 to 2 to 1 ratio. Here, let me get 1 up here. And an example of that would be glucose, a relatively simple carbohydrate, a simple 6-carbon sugar, and its chemical formula is C6H12O6. So it obeys this 1 to 2 to 1, 1 to 1 ratio of carbon to hydrogen to oxygen. Now, carbohydrates play 2 or 3 important roles in body cells. If we put up the next graphic, we'll take a look at some of the functional roles that they play. The simple sugars that have, only typically 5 or 6 carbons in the biological examples, those are energy sources, as well as the 5-carbon sugars are important in nucleic acid structure. If we put a whole lot of simple sugars together, if we put 2 of them together, we have a disaccharide. 3 of them together, we have a trisaccharide. A whole bunch of them, it's called a polysaccharide. Then those kinds of things are very important in cell structure, and receptors in particular are often have polysaccharide components. Lipids. Let's go to our overhead camera for a second. Lipids are insoluble in water. That's their primary physical characteristic. So the other kinds of compounds that we're going to be talking about in biological macromolecules are soluble in water in general. Lipids are insoluble in water. Lipids play important functional roles in cell membrane structure. They're also one form of lipid is an important form of stored energy. So they have very important roles that they play. And if we look at all the lipids in the body, we can actually put them into 4 different groups. Let's take a look at a graphic that will summarize the 4 main subgroups of lipids. The ones that we call glycerides are combinations of fatty acids and glycerol. Let's go to the overhead camera just a minute so I can explain this. Glycerol is a 3-carbon alcohol that has this chemical structure. And if we combine one fatty acid, if we put a fatty acid in this position, actually by dehydration synthesis there would be a H from this and an OH from the fatty acid that would leave to form a molecule of water, and then the rest of the fatty acid would be permanently hooked onto the glycerol root in that manner. Then the resulting compound would be called a monoglyceride. Okay? If we put another fatty acid on in the same manner by dehydration synthesis and hook a second fatty acid on to another of the 3 locations on this glycerol molecule, we call that a diglyceride. And if we put a third fatty acid on, so we'd have fatty acids in all 3 positions on that glycerol backbone, we call the resulting compound a triglyceride. Now it turns out that the triglycerides in the body are commonly called fats and oils and serve as an important storage form, a long-term storage form of energy. The steroids are compounds in the body lipid compounds in the body that have basically 3 hexagons and a pentagon ring form. And some of the hormones are steroids. Cholesterol is a steroid, and they form important components of membrane structure. The compounds that we call phospholipids are any lipid material that has a phosphate group on it. And Particularly in the central nervous system, there are a bunch of phospholipids that are very important in some of the nervous tissue components in those locations. Compounds that we call prostaglandins are lipids that basically consist of long-chain fatty acids that have a five-membered pentagon ring in the middle of their structure. And they are important as chemical regulators in the body. So it's not important that you understand the basic structure or the particulars about it, but just realize that when we talk about a lipid material, we're talking about something that is insoluble in water and that there's four major groups of them. Any questions about any of the things we've talked about so far? Okay, we'll move on to nucleotides. And the reason I want to talk, and let's go to the overhead camera for a minute. The reason I want to talk about a nucleotide for a second is because if we polymerize nucleotides, then we can end up with nucleic acids. But we have to know what a nucleotide is. And a nucleotide has three major components. It has a nitrogenous base. And if you remember from biology, previous biology course, those nitrogenous bases that are utilized in the nucleotides in DNA and RNA are adenine, which we often symbolize with an A, cytosine, which we often symbolize with a C, guanine, thymine, and uracil. And we probably know these as much by the single capital letter acronym as we do by their written out names. If you take that nitrogenous base and hook a 5-carbon sugar on it in the form of ribose or deoxyribose, and then hook a phosphate group onto that, the result is a nucleotide. And so nucleotides are the building blocks or subunits that, when polymerized, form the nucleic acids that we call DNA and RNA. Now, let's take a look at a more formal graphic than the primitive one that I produced here, and we'll take a look at summarize those components up on the screen. The nitrogenous bases, again, are adenine, cytosine, guanine, thymine, and uracil. Now, the two nucleic acids only use 4 of those each, so there's 5 up there, but we'll talk in just a minute the difference between DNA. 4 of those are used in DNA, and 4 of them are used in RNA. The 5-carbon sugars that are involved in the sugar component of a nucleotide is either ribose, if we're going to form the nucleotides that are going to be involved in ribonucleic acid, RNA, or they're deoxyribose, if we're talking about the nucleotides that are going to be involved in DNA, and then the phosphate group, the inorganic phosphate. Now, polymerized nucleotides, if you put 2 of them together, you have a dinucleotide. If you put 3 of them together, you have a trinucleotide. If you put a whole herd of them together by dehydration synthesis, then you end up with a polynucleotide, and eventually, if that gets long enough, we're going to call it DNA or RNA. If we compare DNA to RNA, they have a very similar structure, except there's 2 or 3 differences. Do any of you remember what any of the differences are between DNA and RNA? Susan?
DNA is double-stranded.
DNA is double-stranded, at least in its coiled-up form. It's double-stranded, certainly, and RNA in general is
Single-stranded.
RNA is single-stranded, with the exception of some parts of some transfer RNA, which may have some little double-stranded segments within them, right? So we got a little strandedness difference there. And what's another difference between DNA and RNA? Amber?
DNA uses thymine, whereas RNA uses uracil.
Okay, we talked about there being 5 different nitrogenous bases, and DNA uses 4 of those. RNA uses 4 of them. And there's one obvious base that's different in the 2, and we'll get to that in just a second. That's right on. And the third difference between DNA and RNA, Adam?
Which 5-carbon sugar it uses.
Exactly. I'll tell you, there's not much difference structurally between ribose and deoxyribose. Basically, you take an oxygen atom out of ribose and you end up structurally with deoxyribose. They're both 5-carbon sugars, and the 5-carbon sugar that we find in the nucleotides of ribonucleic acid, of course, is ribose, and the 5-carbon sugar that we find in the nucleotides of DNA is deoxyribose, and that is the basis of the name difference between the two nucleotides. Now, let's go to the overhead graphic for just a minute. Susan mentioned that DNA is double-stranded, and if we have a strand here and we just represent the, let's do it this way. We'll say that's the sugar-carbon backbone, and we have the nucleotides sitting out here, like, or the nitrogenous bases of nucleotides sitting out here in this manner. Another strand would be over here, sugar phosphate backbone, and you'll recall from biology that the nitrogenous base that would mate up with through hydrogen bonding with this cytosine here would be what? Does anybody remember that?
Guanine.
Guanine. In other words, C pairs with G and A pairs with T. So there would be a T here, there would be an A here, and there would be a cytosine down here pairing up with this guanine. And it turns out that there are, in fact, hydrogen bonds that form between these base pairs, and the shapes of the nitrogenous bases are such that cytosine tends to want to pair up with guanine, and adenine wants to pair up with thymine. And in the double-stranded DNA, these are the 4 nitrogenous bases that are found within DNA: C, A, T, and G. Now, let's look at a graphic that'll summarize the comparison of those before we go on. And so we've got 5-carbon sugar is different in the two. RNA uses ribose, DNA uses deoxyribose. The nitrogenous bases that are found in DNA we mentioned are A, C, G, and T. But if we go to RNA, the 4 nitrogenous bases are A, C, G, and U. And the strands that are considered to be present, we said that DNA has the double-stranded form that we just showed, and RNA has single strands. And so there's just a 1 sugar phosphate backbone with the nitrogenous bases sticking off of it. Let's go to the overhead camera for just a moment, and let's take a look at a word that biologists use. What does this word mean in reference to nucleic acids? Yes, Adam?
Creating another copy of itself. Replicating, reproducing DNA.
In other words, to a biologist, this word replication is going to mean using DNA as a pattern to make more DNA, right? Okay. And another word, back to the overhead camera here. Transcription. What does this mean to a biologist? Yes?
When they take DNA and make it into RNA?
It's using DNA as a pattern to form RNA. So basically, the DNA is used as a pattern to form ribonucleic acid. And of course, since ribonucleic acid is a single-stranded molecule and DNA is a double-stranded molecule, what has to happen is the double strands have to unwind or split apart from each other, and only one of those 2 strands is used as the pattern to form RNA. And when transcription occurs, when we do form RNA, what are the kinds of RNA? If I were to ask you what kinds of RNA exist, I know we're going back to some things just to make sure that we're reviewing some biology here.
You know, transfer RNA, messenger RNA, and
Anybody remember the other one? Transfer RNA, messenger RNA, and the one that is involved in the structure atom of ribosomes.
Is
Ribosomal RNA. Let's go back to our overhead camera and let's put those down for everyone. Biologists will call the one kind of RNA transfer RNA. They'll call it little t RNA. The ribosomal RNA that's involved in ribosomal structure they'll call rRNA. And the RNA that is involved in actually carrying the code, if you will, they'll determine what amino acid is inserted in the growing amino acid or polypeptide chain, is called messenger RNA. All three of these kinds of RNA are manufactured in the process of transcription. So the pattern for the formation of all three of those is found in the DNA. And in the process of, again, transcription, all three kinds of DNA are made, okay? And then all three kinds of DNA are involved in protein synthesis. Now, if we go back to the overhead camera one more time, the word that biologists use for protein synthesis is translation. So we want to remember this is protein synthesis. And all three of these kinds of RNA are involved in that process. The transfer RNA actually is involved in hooking up with certain amino acids. Transfer RNAs are amino acid specific, so that means we have to have about 20 different transfer RNAs. They carry the amino acid over to the ribosome. The ribosomal RNA, of course, is involved in ribosomal structure, and the ribosome is a cellular organelle that is involved in the physical process of connecting the amino acids together to form the growing polypeptide chain. And the messenger RNA, mRNA, is the part of the RNA complex that's involved in carrying the sets of three nitrogenous bases that determine what the next particular amino acid to be inserted into the chain will be. It carries the codes, if you will, in. So the original codes, of course, are in the DNA, then they're transferred to the RNA. A codon is, in fact, a three-base sequence on the messenger RNA, and that determines what the next amino acid will be, because there's a different codon for the varying amino acids. I don't know if you've thought about this for a second. If we're taking those nitrogenous bases three at a time, or to form a codon, we got four different nitrogenous bases in RNA, and so that means there's four possibilities for what the first nitrogenous base will be. And then there's four possibilities for the second one, so 4 times 4 is 16. And then there's four possibilities for the third one, and so 4 times 4 times 4 is 64. We have 64 possible codons, but there's only about 20 different amino acids. There are actually some amino acids that have multiple codes that code for the same amino acid, and then there are other codons that actually don't call for an amino acid, but maybe, as you know from biology, start or stop codons within the protein synthesis mechanism. Any questions about that before we go on? I know I just kind of blew through it because it's some history. I kind of wanted to establish a little bit of cytochemistry before we actually start talking about cell structure itself. Anyone have any questions about it? All right. Let's go to the cell itself now and take a look at it from a physical standpoint. We'll take a look at a graphic here that will summarize cell structure. The cell membrane is the semipermeable membrane that surrounds the whole body of the cell. Actually, the fluid that's inside there is called cytosol technically, but if you put the organelles in there within the cytosol, we can call that combination cytoplasm. So cytosol is just the fluid inside the cell membrane. With the organelles, it's cytoplasm. And then I've separated out the nucleus on this graphic because we want to remember the nucleus certainly is an organelle, but it has such important functional roles because it carries the DNA. So perhaps if I were going to do this, it would be better to say cell membrane, cytosol, and nucleus, but many biologists will separate cell components down into these three general areas. So this cell membrane, let's talk about the cell membrane for just a bit. What kind of functional roles does it have? What's important about the cell membrane? Anybody got any ideas? Amber?
Protects the cell.
Protects it it certainly does, but in what way? I mean, it's in what way would you say?
It only allows what needs to get in and what needs to get out.
So it's going to determine what goes in and out to a great degree, is it not?
Yeah.
And that's going to be important. Anything else it does, and it's kind of related to protection in a way, I guess, but by surrounding the whole thing, it's going to do what? Yeah, Adam?
Kind of keeps everything in the same spot.
That's
Keeps
right.
everything together.
Imagine what it'd be like, to try to keep all your organelles and cytosol from dribbling away if you didn't have a cell membrane around the whole thing. So not only does it hold everything together by surrounding the whole thing, but it also controls what enters and leaves, which is critical to the metabolism of the cell. All right, now, how do we get across a membrane? Let's think of ways that we can cross this membrane. I'm some kind of a small chemical entity, and I want inside this cell for some reason. How can I get across there? Anybody? Adam?
Through a carrier protein.
I could be carried across involving, I might have an escort in the form of a carrier protein. Do all chemicals need carriers to get across the membrane?
No.
No. Some things can just go across without a carrier, can they not? So some can go across by themselves, some require a carrier. Any other kinds of ways, Hazel?
Through endocytosis.
Endocytosis is, we're kind of above the chemical level now. We're into a process that's going to take in either a little tiny particle or perhaps a little droplet of something. So we're actually taking in a whole wad of stuff rather than a chemical entity, but that's certainly a way that we can get across the cell membrane. Let's go to the overhead camera and talk about some ways to cross membranes. If we have a carrier, but no energy is required, the process is called facilitated diffusion. If we have a carrier, but we do need energy to get this substance across the membrane, the process is called active transport. If we don't have a carrier and no energy is required, the chemical transport mechanism is called simple diffusion. If water or other solvent is the substance that's involved in simple diffusion, that kind of simple diffusion across a semipermeable membrane is called osmosis. So osmosis is simply, in biology basically, most of the time we're talking about water. It's the simple diffusion of water across a semipermeable membrane. Now, I've said semipermeable. What do I mean when I say this cell membrane is semipermeable? What does that imply? Susan?
That small molecules are certain molecules can go across, but the larger ones cannot.
It'll allow certain things to go across,
right.
and other things not. And usually you're right. Usually it's the smaller things, unless they become extremely hydrated, in which case they're effectively large molecules that may not be able to go across. So semi-permeable basically means some things can cross the membrane and some things can't cross the membrane. Like a doorway in a house. Typical doorway in a house is semi-permeable relative to like elephants and people, right? People can go through the doorway, but elephants basically can't, or shouldn't, at least not at my house. You got the idea?
Right.
Okay. So we've got, the cell membrane is semi-permeable in that it will allow certain compounds to cross and will not allow other compounds to cross. And so that's the reason we call it semi-permeable. Now let's go back to this overhead and take a look one more time of these three ways that we've mentioned here. Water basically crosses by simple diffusion, process that we'd call osmosis. Glucose goes across the cell membrane by facilitated diffusion. Now both simple diffusion and facilitated diffusion only work based on concentration gradients. And the substance will only cross the membrane if it's in higher concentration on this side, in which case it will simple diffuse or cross over to the other side in that manner. You can never use facilitated diffusion or simple diffusion as a way to move a substance from an area of lower concentration of that substance to an area of higher concentration. So for both facilitated diffusion and simple diffusion, the driving force is the concentration gradient, and the diffusion only occurs in the direction of the concentration gradient, from higher concentration of that substance to lower concentration of that substance. On the other hand, this second one that we've put down here, active transport, this mechanism will transport substances from an area of lower concentration, whatever side of the membrane we're on that has less of that substance, over to an area where there may be even more of that substance, from low to high. But it does require, as you can see here, does require energy. Okay? All right, now, these three ways that we've put up here are ways to cross membranes for chemical entities. Now, what Hazel mentioned a while ago, she mentioned a word endocytosis. Let's put that on our overhead so that we can all see it. And some things can enter cells by this mechanism, endocytosis. This basically just means going into a cell, but in practicality, we can break down endocytosis into two different mechanisms. We can break it down into phagocytosis. It's supposed to be an A here. And pinocytosis. Basically what's happening, if you allow this to represent a cell, that's a nucleus, and here's a little droplet or a particle of material that maybe that cell would like to have inside of itself. It can approach this and send out a process of the cell membrane thusly, and eventually completely surround and cut off right here with the cell membrane, taking this foreign substance into it, surrounded by a bit of the original cell membrane. Now, if the foreign substance is a solid particle of something, then the term phagocytosis applies. If the foreign substance that's coming in is a droplet of fluid, then the term pinocytosis applies. So both of these are forms then of endocytosis. Now, cells can move things from within them to the outside by reverse phagocytosis or reverse pinocytosis. So we could do this process in reverse to move a particulate substance or a droplet of a substance from inside the cytoplasm to the outside as well. So these are ways that you can cross a cell membrane that are considerably above the chemical level. Now when you look at ways to cross the membrane, then there's several different ways, and different substances use different mechanisms to get across, and we've already mentioned a few of those, but just wanted you to be aware of how the membrane is involved in what enters the cell and what leaves the cell. Now if you noticed with the phagocytosis that we just demonstrated in a drawing there, there's a little tiny bit of the cell membrane that comes into the cell with each particle or droplet that comes in the endocytotic mechanism. The reverse endocytosis, sometimes called exocytosis, there's actually membrane that's added to the outer cell membrane. So obviously if a cell is involved in this process, there has to be a balance, or else the cell membrane will either be diminished or excess membrane will develop. Any questions about that process? All right, let's go inside of a cell and let's go to the overhead here and list some of the subparts of the cell that we'd find floating around in that cytosol. These subparts of cells, you said the other day, were called organelles. Who knows the name of an organelle and kind of what it does? Seth or Adam?
The mitochondrion produces energy for the cell.
All right, let's put down mitochondrion. Produces energy. What form is that energy in?
ATP.
Okay, so the mitochondrion is involved in ATP formation. That's an acronym that stands for what? Adenosine triphosphate, right? And that's a high energy compound that's used as an energy currency for a lot of the cellular processes, is it not? All right, another organelle. Susan?
I like the Golgi transport, or the Golgi.
Golgi complex. Okay.
And that's involved in the transport of packaging and transport. Is that right?
It is. Golgi complex is involved in packaging, primarily packaging things for exocytosis that are going to be secreted from the cell. Another organelle. Hazel?
Ribosomes for protein synthesis.
All right. Involved in translation, which we said was protein synthesis. Now, it's interesting that some of the ribosomes that are present in the cytoplasm, when they're involved in the protein synthesis mechanism, are actually floating freely within the cytoplasm. And those ribosomes are actually manufacturing proteins that are, for the most part, going to stay within the cell. Okay? Other ribosomes that actually go down and hook onto another organelle called endoplasmic reticulum, we haven't mentioned yet, called membrane-bound ribosomes at that moment that they're hooked onto that endoplasmic reticulum, are going to be manufacturing proteins that, for the most part, are going to be exported from the cell. So we often talk about free ribosomes, if they're just floating freely in the cytoplasm, or we talk about membrane-bound ribosomes. And those are hooked onto the endoplasmic reticulum. Matter of fact, when they're present on that endoplasmic reticulum, we call that endoplasmic reticulum rough endoplasmic reticulum, often acronymed RER. Okay? Another organelle. What's another important organelle that we could list? Seth?
Lysosomes?
Lysosomes.
Those contain digestive enzymes and things
They
like
do. Lysosomes contain hydrolytic enzymes that are involved in breaking down components within the cell, involved in hydrolysis of things within the cell. We better put up endoplasmic reticulum since we've already mentioned it. ER stands for endoplasmic reticulum, and that can either be smooth if it has no ribosomes on it at the moment, and we acronym that SER, or it can be rough if it has ribosomes on it at the moment, and we acronym that RER. Now we could go on, but there are a whole lot of other organelles as well, and we're not going to list them all. But the idea is that cells contain numerous subparts called organelles. Each one has a specific functional role within the cell, and we call again, organelle meaning little tiny organ is the implication. Let's take a look at a graphic that summarizes the organelles that we just talked about. Ribosomes, free and membrane bound, endoplasmic reticulum, the two forms of it, mitochondria, ribosomes. We didn't talk about centrioles. Centrioles are actually granular structures that are involved in organizing the spindle fibers during cellular division, which is the next thing that we want to talk about. We did mention the Golgi complex involved in packaging things for export. Let's talk just a little bit about the cell cycle in the time that we have remaining. The cell cycle, I think it's important to remember that some cells divide every few hours. Certain body cells divide every few hours. Some body cells divide every once in a while. And once we reach maturity, certain body cells never divide again. Among those would be neurons, skeletal muscle cells. Once we're mature, those cells are done. Some of the cells that divide extremely often are some of the lining cells of the alimentary canal. Small intestine and large intestine in particular have very high rates of cellular division involved in the cells therein. The cell cycle is divided into 4 phases. Let's look at a graphic that will summarize these very quickly for us. The G1 is called the first growth phase. S symbolizes what's called synthesis, and that's where the DNA is replicated. The G2 phase is the second growth phase, and the M phase of the cell cycle stands for mitosis. Now, mitosis symbolizes or is used to represent the regular division of the nucleus involved in forming regular body cells. Does anybody remember the term, and we'll talk about this later when we talk about the reproductive system, but what's the term for the nuclear divisions that are involved in forming sex cells? Adam?
Meiosis.
Meiosis. So mitosis is regular nuclear division to form regular body cells, and if we're going to form sex cells where we have to reduce the chromosome complement, the process is called meiosis. Now, mitosis, just to review what you learned in a previous course in biology, mitosis is actually divided into 4 major phases. And does anybody remember those phases in order? Anybody? Hazel?
Prophase.
Prophase.
Yeah. Metaphase.
Metaphase.
Anaphase.
Anaphase.
And telophase.
And telophase. Prophase, metaphase, anaphase, and telophase. Let's put those up on a graphic so everyone can see the words. Subdivisions of mitosis. Those divisions, which Hazel mentioned, again, are prophase. What's happening here is the cell nuclear membrane disappears and the chromosomes become visible. In metaphase, the chromosomes line up in the middle of the cell at what's called the equatorial plate. In anaphase, the sister chromatids of each chromosome separate from each other, and one sister chromatid goes to one pole of the cell, and the other one is pulled by the spindle apparatus to the opposite or opposing pole of the cell. And finally, in telophase, the 2 nuclei separate from each other, nuclear membranes form, and then finally, you see in parentheses there, cytokinesis, not technically part of the mitosis division process, is when the cell membrane comes in and separates the cytoplasm and the remaining organelles into 2 components. So what we end up with here is 2 completely separate cells that have the same genetic complement. Both of them have a full set of chromosomes, and that's how cells divide, or how they multiply by dividing, if you will. Let's go to our overhead camera a little bit and talk about some clinical considerations where cells are involved, some clinical conditions. When we talk about atrophy, which can happen to cells, it can, of course, happen to organs as a whole as well. Does anybody know what atrophy means? Susan?
That means when they get smaller. Or, for example, skeletal muscle cells, you lose.
Okay, it's a loss of size and/or functional ability,
Right.
is it not? And that can be caused by disease or disuse, or maybe even senility can cause atrophy. How about hypertrophy? What does this term mean? Who wants to define it for us? Adam?
It's the opposite of atrophy. It's growth in size or hyperuse, isn't it?
It's an enlargement of cellular enlargement, or it can happen to a whole organ as well. So it's basically, as Adam says, it's the opposite. And hyperplasia is another term that we probably ought to know. I'll define this one real quickly here. This is an increase in cell number. Now, you realize that an organ can undergo hypertrophy by either cellular hypertrophy or cellular hyperplasia. Either one of these at the cellular level can produce hypertrophy at the organ level. Does that make sense to you? Metaplasia. If a cell changes its functional role, we say that cell underwent metaplasia. If the new functional role that it assumes is more specific than the original functional role, we call that process differentiation. So these two are very closely related, but if it becomes more spatialized by the process of metaplasia, then we call that process differentiation. Mutation is a change in cellular form because what changed? The DNA. The DNA. So if the DNA changed, we say, hey, we've got a mutation. There's a lot of different things that can cause that as well. Chromosomal aberrations are important. Anybody know any particular condition that's related to a chromosomal aberration within cells? Adam?
Down syndrome?
Down syndrome is one that most all of us are familiar with. And when we get cells that are multiplying out of control, what do we call that condition?
Cancer.
Cancer. And what's the 25-cent word for cancer?
Neoplasia.
Neoplasia. You bet. Okay, good.